Hi, I am Dr. Jatinder Singh Chera and I am a PhD in animal biotechnology. And I'm currently working in the field of bioinformatics, which deals with analysis of large data sets and analyzing them for uh, getting some meaningful biological output. Now, to demonstrate the data analysis which I have been using in my various projects, I will just brief you about what I'm going to show you in this video. Here, there is a site called RCSB PDB, which contains lot of information regarding to the structures of biological molecules. Now, for this particular video, I'm going to show you how I obtain different structures from the PDB website, the details of those structures, and how I use them for my data analysis. So, to brief you, I have two files here. You can see .text files. One is na underscore type and one is annotations.txt. Now I downloaded this from some other website, and you can see here I have data related to PDB IDs, polyclass and methods. Now these are the IDs which are available in the PDB website, and these correspond to unique biological structures. For example, let me show you. Like we search 100D, which was on the first number of the text file. So here you can see this is some crystal structure and whatever it is. So these are all the IDs of different PDB structures. Now what I want to do here is that under the column poly class, I have many different kinds of molecules like DNA. There is also RNA. You can see here. Here it is RNA, RNA. There are some protein DNA hybrid, protein RNA hybrid also, like here. So basically what I want to do is I want to take out all these sequences first, uh, sort them and create a different Excel files for each polyclass type. For example, separate CSV file for DNA, separate for RNA, so that I have clarity on what I have and what not in those files. Now there is another file named annotations. Here I have similar PDB IDs but with some additional details. For example, I don't want the file PDB IDs which contain these terms double helix, or you can see here there is triple helix also here and here also. So there are three terms that I don't require. One is double helix, triple helix and parallel helix. Now what I will do is that I will be showing you the running of two scripts that I wrote for this purpose. One is sort.py and one is pdb_download.py. Sort.py will first take the input of these two files, the na type and annotations. Firstly, it will remove the IDs which are containing the double helix, triple helix and parallel helix terms, and then sort all the IDs in the NA type text based on this polyclass. So let me show you how this works. I'm going to use Windows subsystem for Linux for doing my programming. So we will just navigate to our desktop folder in which these files are lying. So now these are written in Python language. So we will just run this script python sort.py. Now I have also modified the script in this way that it will show me what it is doing in real time. It says it found these many entries in the which have unwanted motifs like those that I mentioned, triple helix, double helix and parallel helix. It returned these many things. Now what it has done, it has created a folder named output, you can see here, and within that separate folders for each polyclass have been created so that I get only those CSV files that have data for one particular polyclass. Now in the last step also I have also written a verification code which verifies that whether the files have been written correctly or not. So it is matching the entries again. Whatever is written, it is matching with the master file. So now I'll show you the output folder, and for this particular example we'll be using the DNA underscore RNA.csv file, which is a separate polyclass. Here you can see it has first created a master file from which it has created all these folders. And it has also created an excluded IDs to check what have been excluded here. For example, if I take this 122D, let me search it in the PDB website. 122D. Okay, so you can see here, this is the DNA helix. It's a double helical structure, and based on the details which we had in annotations, it removed this sequence because it is a double helix. So now taking this example DNA underscore RNA, you can see from the na underscore type text file, it picked these many IDs which fell in this polyclass DNA slash RNA. So what I am going to do now is that using these IDs, I need to download the PDB structure files from this website. Let me just show you what I want to download. For example, let me search this one BR3. Now here there is an option to download files. I am interested in this file, PDB XMCIF format. But there are so many IDs here. So I have automated this script for doing this task. Now let me show you first. Here I have a script name pdb underscore download.py. So what it will do, it will read this Excel file and download all the IDs that are mentioned here. Now in this script, I also have an option to do batch downloading, which is that it will parse all the folders for the CSV files and then download all the PDBs mentioned here. Like for example, in DNA, these many IDs are there. So manually downloading them is very tedious task. So what I have done is I've written this script and it will download all the files automatically. But for this particular example, we'll be just focusing on the DNA slash RNA CSV. So what I'll do is I'll write Python pdb underscore download dot py, and I have to give an argument here which is to download a specific folder, a specific CSV in a specific folder. So you can see here my directory name is DNA underscore RNA. So oops, sorry. Let me see. DNA underscore RNA slash DNA underscore RNA dot CSV. Sorry, by mistake I did one slash. Now it is downloading all the entries. So all the downloads have been completed for this particular CSV file. These files have been downloaded. Within that folder, there is a file called structures. Now, these are the PDB files that are required. And to show you one example, we'll just check this. This one BR3. You can see some details here. We'll match that after searching. One BR3 crystal structure of 82 nucleotide. You can see it is the same file. We can search one more, one EGK. One. So we'll just check another PDB file that we downloaded. You can see crystal structure of nucleic acid four-way junction. It's the same file. So by this method, we can automate this data writing, first data fetching, then data writing into the Excel sheets and separating them, categorizing and organizing them, and ultimately doing analysis related to these files. So that's all I wanted to show in the video. And if you're looking for someone reliable and efficient and detail oriented in regards to data writing and analysis, I'd be happy to help you in your next project. Thank you.